Hello students, today again I am back with the topic on transcription. Transcription, which is the process in which an RNA is produced, that is ribonucleic acid is produced from a DNA. The codes of DNA are copied onto the RNA and thereby the RNA that is prepared will be carrying out the different functions like translation, that is formation of proteins, or it will assist in the transfer of amino acids, or it will go and attach with the ribosome, or it will become a part of the ribosome. So today, now we will be discussing or trying to learn how the transcription process starts and how it is completed. Now, transcription, once more, in transcription is the process of copying genetic information from DNA to RNA. The genetic information, that is the codes that are present in DNA, which we have already learned in our previous lectures or previous classes. Now, the codes that are present in DNA, they will be copied to form a RNA, that is the single-stranded RNA. And the formation of RNA, just as DNA replication takes place in the direction of 5 to 3, similarly, formation of RNA also takes place in the direction of 5 to 3. The enzymes mainly responsible for the process of transcription is DNA-dependent RNA polymerase. Some additional factors are also required which will help the RNA polymerase to function properly. The additional factors sigma and rho. Sigma will help in starting the process and rho factor will help in ending the process of transcription. And in transcription, during the process of transcription, the RNA formation will take place with the help of only one DNA strand. Out of both the strands of DNA in a double helical structure, only one of the DNA will be copied to form the RNA. Both the strands are never copied. Only one of them will be copied, and the one that is copied and the one to whom the RNA will be formed with complementary bases is called as the template strand. The other strand to which the new RNA will be exactly similar, the bases will be exactly similar, that is known as the coding strand. Then mainly three types of RNA are transcribed or are formed, that is messenger RNA, mRNA, transfer RNA, that is tRNA, and ribosomal RNA, that is rRNA. mRNA functions in transferring the codes of DNA to form the new protein. That is, messenger RNA will act as a messenger of the type of DNA or the type of sequence of bases in the DNA that needs to undergo translation or formation of protein. Transfer RNA helps in transfer of amino acids during the formation of proteins. Ribosomal RNA is a part of the ribosomes that is responsible for protein synthesis. Now, both the strands of DNA are never copied. Why? Because if both the strands would code for proteins, then it will result in one of the segment of DNA coding for two different proteins. That is, both the RNA that will be formed out of the DNA, if it would have happened, then both of them will start the process of translation independently and both will produce proteins from the same strand of DNA, which will be quite confusing, which will not be able to express in the form of proper Mendelian factors, of proper Mendelian characters. Or also, if in case both the DNA strands undergo transcription, then two RNAs will be produced, where again those two will also be complementary to each other. Thereby it might result in the formation of a double strand, which is just next to impossible. That will not be an RNA. Now let us go to the transcription unit. Now what is a transcription unit? Transcription unit is the part of DNA that will undergo transcription. It's the part of DNA that will undergo transcription, that will be responsible for formation of the RNA. Now the transcription unit, that is in a long strand of DNA. In a long strand of DNA, suppose this is the DNA, in a long strand of DNA, now if only this small portion of DNA is undergoing transcription, is responsible for formation of RNA, that portion will be referred to as the transcription unit. Now, what will define a part of DNA to be the transcription unit? The part of DNA which will have one promoter region, one structural region, and one terminator region will be defined as the transcription unit. Now, the promoter unit, that means this particular portion of DNA will be made up of bases upon which the RNA polymerase, the enzyme responsible for transcription, can bind and start with the process of transcription. Following it is the structural gene. The basis upon this portion, the portion of the structural gene, the codes upon it, will be responsible for the formation of RNA. So the RNA transcribed, RNA formed will be out of the bases that are present within the structural gene. Finally, the terminator region will be again made up of some bases upon which the polymerase cannot bind anymore. So, on reaching the terminator region, the polymerase will automatically move out. It will move out. Of course, there are other factors involved there. It will move out. So, the transcription unit compulsorily should be having a promoter region to which the polymerase will bind, structural region which will undergo transcription, and the terminator region which will terminate or end the process. Other than that, the two strands of DNA which are complementary to each other, one of them will behave as a template strand and the other will behave as the coding strand, as already mentioned previously. Now, as the formation can take place in the direction of 5 to 3, so depending upon the position of the promoter, the template strand can be determined. Like say the promoter is present here, which means the transcription, the polymerase will bind here, so obviously transcription will take place in the direction of the arrow that is shown here. So it will take place in this direction. So if it has to take place in this direction, as we already know, formation takes place in 5 to 3. So the new RNA will be 5 to 3 formed in the here. It will be formed 5 to 3. Now, so basing on the position of the promoter, we can assume, or we can easily define the strand which has the polarity 3 to 5 with reference to promoter will be behaving as the template strand, and the complementary one will behave as the coding strand. The three parts that makes up the transcription unit: promoter, binding site for RNA polymerase; structural, polymerization of RNA will take place with the structural; and terminator, end of transcription. Now the stages involved in the process of transcription: initiation, elongation, and termination. In the first step, the very first step, what will happen? The promoter region as already defined. So here we have the promoter, the structural, and then the terminator is present in the DNA helix. Now, the red strand that is shown here, this is 3 to 5, and the blue strand is 5 to 3. So here, as the red strand is 3 to 5 and promoter is present at this end, okay, so what will happen? The formation of RNA will take place from left to right with reference to the picture given here. So the red strand that is shown here, it will act as the template. Now, RNA polymerase, DNA dependent RNA polymerase comes and binds upon the promoter region when transcription has to start. So it will bind with the promoter, and a factor which is known as sigma factor will also bind with the polymerase. It binds with the polymerase and then it will start proceeding. It will start proceeding along the length of DNA. As it proceeds, what happens? The DNA opens up. As we can see here, the DNA opens up. So here, this is the first step, that is binding of the polymerase with the sigma factor. Second step, that is initiation, or rather we can consider this as the first step only, initiation. So in initiation, what will happen? The polymerase will open up the hydrogen bond only at a certain portion, not the entire DNA. As it moves, the nucleosides, ribonucleosides complementary to the template strand. This is the template strand. So complementary to the template strand. If here was present A, then the complementary strand in the complementary base in RNA will be U. So a nucleoside with base U will come and take position. If suppose the next base, DNA base, is T, thymine, then the complementary base for RNA, it will be adenine. So U first lined up, then A will line up. Suppose here it was G, the DNA had guanine, then the complementary base C will line up. If the fourth base was C, cytosine in case of DNA, then guanine will line up for RNA. So one by one they will line up here. As they line up, also there will be formation of phosphodiester bonds. So phosphodiester, so here is the RNA which was already formed, or which has already been formed. Once the RNA starts forming, it will gradually slip away from the DNA. It will not remain attached to the DNA, it will slip away from the DNA. Polymerase proceeds. As polymerase proceeds, more and more bases are added. As the polymerase proceeds, the new RNA moves out. The other side, the DNA will again go back to its original position. So it proceeds, and simultaneously that side sigma also moves out when it approaches the stop sequence or the terminator region. Elongation. So initiation followed by elongation, which I have already mentioned. Then finally, when the stop sequence it reaches, when the polymerase reaches the stop sequence or the terminator region, the polymerase will move out. Which will be assisted by a factor called Rho. Rho factor will assist in moving out of the polymerase. So thereby our RNA formation is complete. So the first step, initiation, binding of RNA polymerase on promoter and association with sigma factor to initiate the process. Polymeration of nucleoside triphosphates. Then the third, termination, association of Rho factor with polymerase and giving out of the new RNA. Now, in prokaryotes, there is no complexity. In prokaryotes, the RNA that will be formed can straight away start with translation process once it binds with the, or it gets associated with the ribosome. But in eukaryotes, it does not happen so. In eukaryotes, what happens? There are some additional complexities. Now let us see those complexities. In eukaryotes, there are three types of polymerases. RNA polymerases, the enzyme, there are 3 types. Unlike prokaryotes, where we find only one type of RNA polymerase responsible for formation of RNA. But here, unlike the prokaryotes, there is RNA polymerase 1, 2 and 3. Each of them responsible for formation of different types of RNA. Polymerase 1 will help in formation of ribosomal RNA. 2 will help in formation of heterogeneous nuclear RNA. Now, what is this heterogeneous nuclear RNA? The one which we have just now seen, the formation, that is heterogeneous nuclear RNA. Heterogeneous means mixture, nuclear RNA. So the RNA that is formed will be made up of coding and non-coding sequences. Hence, it is called as heterogeneous nuclear RNA. But the cell requires only coding regions. So what will happen? The RNA, hnRNA, has to undergo some additional process so that finally it will go to form the messenger RNA. RNA polymerase 2 responsible for formation of hnRNA, which later modifies to form the messenger RNA. Polymerase 3 formation of transfer RNA, then 5 small ribosomal RNA, small nuclear RNA. hnRNA, as I mentioned, it has coding and non-coding regions. Exons are the coding regions, introns are the non-coding regions. Furthermore, in prokaryotes, we have polycistronic genes. Polycistronic means, cistrone means the gene when it is coding for a polypeptide. When the gene, when it is expressing in the form of a protein, then it is called as cistrone. When it is a unit of protein formation, we call it as cistrone. Poly means many. So, in prokaryotes, a single gene, from a single gene, one RNA is produced. That RNA can go to form, that RNA can go to form the different types of proteins which will be involved in the metabolic pathways. Single gene, many proteins. But in eukaryotes, in higher organisms, that is not the case. In this, what will happen? The single gene, single protein. So, it is referred to as monocistronic. Now, we will see how the hnRNA modifies or changes to form the messenger RNA. Now, this is the structure diagrammatic representation of pre-mRNA. Pre-mRNA means hnRNA. The RNA that we saw little while ago, we saw how it was formed during the process of transcription. Now, the pre-mRNA, pre-messenger RNA or hnRNA, it has 5' end, it has 3' end. It is made up of bases. Here we are not showing the bases. Alternately, it is made up of some coding region and non-coding region. Coding region, non-coding region means there's only the portions which have been marked red here, they are responsible or they can help in protein formation. They can help in coding for the protein. Whereas the portion here that is marked light blue, they are not responsible or they cannot code for anything. They are not expressed. They cannot be expressed in any form. They cannot be expressed during the protein formation. They cannot code for any amino acids. This can be related with euchromatin and heterochromatin, which we had learnt in the previous quite few classes back. We had learnt like during the packaging of DNA. They can be related with heterochromatin and euchromatin, transcriptionally active and transcriptionally inactive genes. So exons are transcriptionally active or coding regions. Introns are transcriptionally inactive or non-coding regions. Now, what is required during the processing of hnRNA? It is required that these are removed. because it will be an unnecessary expenditure of energy on the part of the cell to transport the whole of the mRNA, whole of the hnRNA out to the cytoplasm, and apart from that, it will not be able to form a proper protein. There will be breaks in it due to the presence of those known coding regions. So it needs to be eliminated out. So that process involves splicing. Now what happens in splicing? The introns are removed. An enzyme called spliceozyme will act and cut the introns at the ends. The ends will be cut off, and consequently the introns will be removed out. Then again enzymes will play the role to rejoin only the exons. So that step is known as splicing, following which there will be capping. The 5' end. The 5' end, there will be addition of compound methyl guanosine triphosphate here at the 5' end. It will be added. That step is known as capping, followed by tailing. In tailing, what will happen? Many adenines or adenine-led residues will add up. Many AAA will add up at the 3' end, forming a tail-like structure. So that is called as tailing. So the final mRNA that we get will appear somewhat like this. Diagrammatic view. Only exons, introns have been removed with the cap at one end and the poly A tail at the other end. So this will be the final mRNA structure after processing of hnRNA. Now let us have a look at one more slide how splicing, capping and tailing actually takes place. So here we have a figure as given in a textbook. So this is the DNA. The first is the DNA which is undergoing transcription. The polymerase shown here, it is undergoing transcription, and we have got here the hnRNA. So hnRNA, what will happen to hnRNA, which is made up of exons and introns? It is made up of exons. The yellow ones are exons and the red ones are the introns. So what will happen? Splicing. Splicing will take place, and the process of splicing, what will happen? The introns will be detached. As we can see here, this portion has been cut off, and you get only, or we get only a shorter strand of RNA containing only exons. Then other than that, the additional steps are capping. So at the 5' end, there will be addition of methyl guanosine triphosphates, so which will be added to the 5' end, called as capping, and at the tail end there will be addition of poly A tail, which is known as tailing. So the final structure, you will get methyl guanosine triphosphate at 5' end, only exons, and then we will get the poly A or many adenylate residues at the tail end. So this completes the process of transcription. Once this is done, once the process of mRNA formation is completed, then mRNA will then be transported out of the nucleus. When it is a eukaryotic cell, here we are talking about eukaryotic cell, it will be transported out of the nucleus through the nuclear pores to move to the cytoplasm. In the cytoplasm, it will attach with the ribosomes, which might be free or which might be attached with the endoplasmic reticulum. They will attach upon it, and then it will start with the translation process. Translation process, which is the formation of protein. So in the next video or next lecture, we will talk about the process of translation. So do watch the next lecture, so that the continuation of your the understanding the concept remains.